He's a professor of physiology and biophysics with Weill Cornell Medicine and the director of its bioinformatics core in Doha, Qatar. Let's welcome Dr. Suhre.
Yes, hello, hi. Thank you very much for the nice invitation. And it's really sad that I couldn't be coming by in person, but maybe you can do that another time. So I will be talking today about genetics meets proteomics, perspectives for large-scale population-based studies. What this actually means, I will be talking about what we did in the field of GWAS with proteomics. So as I'm from American University, I have to have a conflict of interest slide, so that's here. For those who don't know where I'm coming from, so I'm working for Weill Cornell Medicine, which is down the road of Mount Sinai, but the Qatar campus in Doha. And what you see here is our building. So in the middle is the lecture halls. There is on one side, you have all the research buildings, and the other is the teaching. And we actually train about 40 to 50 medical doctors every year who get a full Weill Cornell Medicine diploma and then go on to training and to matching in the US. So you may even have met some of our students in Mount Sinai. All right, so just to show you where we are, it's somewhere here on the side of Saudi Arabia, opposite to Iran. Okay, and why I'm working on this is, so Qatar has a pretty high diabetes rate, and they invested a lot of money into getting independent of oil and to solve their own problems. And as part of this, they invited Weill Cornell to Qatar to do teaching and research in medicine, especially in diabetes. So the central question here is, I mean, I should mention also in Qatar has about a diabetes rate between 20 and 25%. So diabetes is a very, very big question here, and we want to understand what actually makes us different. And just as a quick introductory slide, I mean, of course, you probably know most of this. What defines us is our genes, our lifestyle, what we eat, what we drink, whether we're physically active, the drugs we take, and then, of course, also our gut environment. And all this is what we want to study in order to understand the different diseases. And as I already said, the most central one for Qatar is diabetes, but of course, other diseases are also very prevalent here, especially cardiovascular diseases. So as a bioinformatician, we work on a lot of omics data, and what my group has been working on the last 10 years is integrating different omics phenotypes. So there's, of course, the paradigm of biology from gene to RNA, from DNA to RNA to protein, translating into genomics, transcriptomics, proteomics. But I always like to extend that to the effector molecules, the metabolites, the metabolomics. So I won't be talking about this today, but before going into the proteomics field, we did a lot of GWAS on metabolomics as well. And at the sideshow, we also have now looked into epigenome-wide studies, microRNAs, and also protein modifications, especially glycomics. So all these are omics technologies that you can study on large population studies. And I will focus on proteomics here. And the reason is, one of that is we have had a few years back a SomaLogic platform here in Qatar, which is a high throughput affinity proteomics platform. And I will be explaining this to start with. So the idea is you want to measure as many proteins in a blood or in a biosample as you can. And the SomaLogic technology is actually what is called affinity based. So opposed to mass spec, where you just count the different peptides you find in the sample, affinity-based is a bit like antibodies, simply that they don't use antibodies, but they use aptamers, which are like these kind of DNA molecules, modified DNA molecules that are in a complex technology fitted into targeting very specific proteins. So what you see here is protein structures, for instance, IL-6 in complex with a SOMAmer aptamer. So how do they convert this kind of technology into measurements? So I would like to go into this a little bit more detail, because I think it's always good to get a feeling for what kind of data you're working with. So this is taken from the SomaLogic's white papers. So what you start off with is there will be SOMAmers. These are the pink ones. They are linked via a fluorophore, a photocleavable linker, and biotin to streptavidin beads. So they're immobilized on these beads. And then they are incubated with the sample. So you take your blood sample, you pre-treat that, of course, as well, and then you incubate that. And what then happens is that the aptamers bind to the proteins and they form complexes. And these complexes can be cognate ones, or non-coordinate ones. So some which are really specific and non-specific ones. So the beads are then washed to remove the unbound protein. Then the bound proteins are tagged with biotin. So they have here the extra biotin going on. And then they are photocleaved. That's why there's a photocleavable linker. So you cleave them off these beads. And then there is disruption of non-specific interactions by specific buffers. And then you capture them again on the other biotin side on a new set of streptolin beads. And then with more washing, you actually wash away all the non-specific bindings. So in the end, you just have the aptamers bound that are really specific. So you actually translate protein concentration into aptamer concentration. So you denaturate these, take off the aptamers, and then you capture them. And actually, this is then just like DNA molecules. You can put them on the microarray chip. You could theoretically also do it with sequencing. And you now have converted protein concentrations into DNA concentration that you could then measure just like normal microarrays. And then you quantify them by fluorescence. And then one point is calibration. So what SomaLogic does, they have a number of calibrators. So each color here is a separate plate on which they have different calibrators. And this is the distribution of random fluorescence units. And to the left, you have just the distribution of the blinded size. So this is just a normal curve. And then they calibrate the calibrators to, so I can maybe flip this around, before and after calibration, so that actually all the calibrators go on a single line. And what you see is that the samples which are not touched by the calibration all come together as well. That's a good proof that this calibration is really working well. And then if you go for CV, like reproducibility of the measurements, so what they have shown is that you have like the variance, the technical variance in the data is, now you have to think about there are thousands of aptamers. So about for 50% of the aptamers, the CV is below 5%. For lower ones, it's even lower. And 95% of the measurements actually have a CV below 12.6%. And that's data, I have this from their white paper, what we have obtained this data in our studies as well. So this is pretty reliable and very reproducible. An extra thing they do, so in blood, one problem in blood is proteomics is that you have a huge dynamic range, around 8 orders of magnitudes between different proteins. And one trick they have, they actually dilute the samples in 3 different bins, like 40% dilution, 1%, and 0.005%. And then they expand 3 times. You see the numbers here, so they don't use the same aptamers in the different dilution bins. So there are different numbers here. Then later they combine them. So you would have like the best dynamic range of 10 to the 8 by doing this. Another thing important to know is, I mean, these aptamers are actually, it's like in metabolomics targeted. So you're not looking for anything, but you're looking only for the ones you know already. That would be like just like antibodies. You can only measure where you have already an aptamer for. So the panel of aptamers that's on the SomaLogic array is of course biased towards what they have developed and what they have trained. And you see here with the distribution. So you see here, for instance, there's a lot of growth factors, receptors, cytokines, kinases, and other kind of things. Proteins are secreted. There's some intracellular ones. I come into a moment why intracellular when you look into blood, and extracellular domains. And also the interest of the protein is a bit varying. You see there's a lot of cancer. I have never really gotten an explicit answer from SomaLogic on this, but I think it comes historically also from what they are interested in their studies. So there's a bias on all kind of things. So you have to be careful later on. If you do your analysis, you cannot just do simple enrichment studies on this. So that would be biased if you don't consider the background. All right, so to wrap these things up, so what we measure, and I think I said that before, we measure proteins in blood, but what does it really mean to measure proteins in blood? That is what I try to display here. So actually you could have proteins like AST and ALT, these liver proteins, they are actually leakage proteins. I just draw them here at the side of the bloodstream, but they could come from the liver, they could come from everywhere. So there could be proteins that actually end up in the bloodstream, even if they are supposed to be there. And that's why that would be the intracellular proteins that are on the panel. Extracellular domains, they are proteins like IL-6 receptor, which are actually cleaved. So they would be on cells at a certain time, and then they would be cleaved, and you would aptamine they actually targeted and also different forms. So you have to be aware also there are different. It's not like a gene. There's not just one sequence. There could be even different forms of the same protein, like a cleaved form, a non-cleaved form, a modified form, things like that. Then, of course, there are proteins that are actively secreted, like cytokines. And then you have proteins that actually come with the blood cells, proteins that are maybe on the surface of white blood cells and things like that. So you also have to be careful about confounding. So you would have, of course, confounding with the different blood cell types. And then what happens is you capture all this, and then you put it through the SomaLogic platform, and then you would get quantitative readouts for each of the ones that are. So at the moment, SomaLogic, not at the moment, but the platform we use targeted between 1100 and the next version, 1300 proteins. So with this, we now go into our analysis. And I think there's nothing, a word I never know who came up with this, but actually large population cohort studies, like using biobanks, UK Biobank, all of us. I think the idea behind this is actually like experimentation on humans. It's experiments conducted by nature. So each one of us is actually a realization of a combination of genes, of lifestyle, of everything. And by measuring large population cohorts, it's just like measuring a lot of different, I mean, we're not lab animals, we're not monogenic, that's why you need big numbers. But it's like experiments conducted by nature. And that's what I really like in the way of studies, in these population-based studies, where you then combine all kind of phenotype to all kinds of endpoints. And I mean, you very certainly know about the Framingham study. I think that's basically the mother of all these kind of population studies, where you would have, of course, genetic information, epigenetic as well, microbiome you might have, and then all this kind of questionnaire data and clinical biochemistry data that you could then link to endpoints and see, is smoking giving us cardiovascular disease and so on. That has been done for tens of years already. But these population-based studies actually identify risk factors, and they're good at that. But they have more problem in revealing really causal pathways and novel biomedical understanding and things like that. So the idea, and I think initially that came more with the metabolomics field, was to put intermediate phenotypes into the middle. So to say deep molecular phenotypes, I know call them, or some people say multi-omics, like big measures for epigenome, transcriptome, and so on. And then you can associate these in GWAS, in microbiome association studies, PHEWAS, phenome-wide association studies. And then, of course, you can also do MWAS, PWAS, proteome-wide association studies with these endpoints. And you can associate each other to each other. And I think that's really something bioinformatician assistants, biologists like. I mean, a really rich way of analyzing that. And taken together, I think the paradigm is that these deep molecular phenotypes, sometimes intermediate phenotypes, reveal affected pathways and can indicate therapy options. And I think there's a lot more Mendelian randomization, polygenic risk scores. So that as terms of introduction, and tell you what kind of data we talk about. And now I want to focus on this genome-wide association to proteomics. So that's something we studied. So this is Johannes Graumann. So he's originally from Germany. He was here in Qatar for 5 years, and he built up the proteomics core in Qatar. And he was the person who brought the SomaLogic platform to Qatar. And we partnered together, put our remaining startup money to actually measure SomaLogic data in a German cohort and in a cohort I collected here in Qatar, and then did this GWAS study. And this is now what I want to talk about. I mean, in the meantime, there are some bigger studies, but in the end, everything from the concept holds for the bigger studies as well. So I think it's easy to go through this. So GWAS with proteomics. So what we did, I think I mentioned already, we put our money together to get these samples measured. So we measured 1,000 study samples from the CORA study. So CORA is basically the German Framingham. We had the platform with the 1100 proteins at the time. We had AFI axiom SNP data on the CORA study. And then we ran linear additive genetic regression models using Blink with the normal covariates, age, gender, BMI. And then we used inverse normal scaled protein levels. So you just rank scale them and put them onto a normal scale curve and ran the GWAS on them. So maybe just, I don't know how much you people are working on GWAS, but I mean, there are a few things. For instance, we didn't use imputing here. We just worked with a AFI arrays. I mean, in the meantime, people have done a lot of more stuff with imputed data. You could, of course, throw in much more covariates here. And I think the central question is also, what do you do with the scaling? Because if you have 1,000 proteins, it's very hard to do the QC on each and every of them. So going for inverse normal scaling, I think it has become a bit of a standard. It's statistically not the most powerful method, but what it does, it kills all the false positive signals. So if you have like 2 or 3 outliers that just go with 1 or 2 rare variants, you would get like very, very low p-values, which in most cases you would not replicate, so you couldn't do anything about them. Not meaning that they might not be true positives. So here's the Manhattan plot. And I think this is a bit like already what we've seen in metabolomics, are really extremely dense. Now this is a Manhattan plot, of course, of all 1,000 proteins together. That's why it's so dense. But what you see, there's tons of signals, and it's just plateaued here at 10 to the minus 20. The p-values go into the hundreds, and I've seen colleagues now even getting into the thousands. So actually, each of these genetic loci look like this. So this is the genetic, the regional association plot, where you would have the chromosome position. Here's the protein ERAP, for instance, and this is the aptamer targeting ERAP. So this would actually be what I would call a cis pQTL. So a protein quantitative trait locus located in cis, meaning that the gene encoding the protein is at the same locus. And of this one, we have many of them. And then you can also look at the top hit, for instance. So if I take here the strongest association, they are so strong that you can even draw box plots of them and get. So here's the p-value 10 to the minus 126 at this case. You have here the different alleles and the count of the allele counts. So this is a very, very strong association with ERAP1. The gene name is ARS1. So altogether, if I look now at Bonferroni level, so we want to be conservative because we have so many hits, so it can be strict here so that we don't get too many false positives. So if I correct for the number of SNPs and the number of tested proteins, my Bonferroni level would be 10 to the minus 11 times 8. And at that level, we find 539 pQTLs, meaning a genetic variant associated with a protein and pruned for everything down to an LD of R square of 0.1. So only counting individual segments. This is for 2,284 proteins. So remember, we measured 1,100. So it's about a little bit about a quarter, a third of the proteins that have a pQTL at the power we have in our study. And independent loci. So at some loci, you have multiple protein QTLs. So there are 451 independent loci. And so this is the summary. And then we can try to replicate this. So I said we have another study here in Qatar where we had 338 participants. So you see again the numbers compared to other GWAS, they're pretty small, but the effect sizes are so big that even with that numbers you can do something with proteomics. So for replication, of course, you need to find a matching SNP, so that reduces a bit the number of loci that we can test. So of these 462 for which we actually had a tagging SNP, half of them replicated at Bonferroni level for replication, and a few more were significant. But remember, we had a study which was more powered than QM-Diab. So if I now only look at the association that actually I would expect to be powered enough to replicate in the study of 338, we only have 208 to look at with 95% replication power. And of these, we replicate 82%, and nominal they're even 95. So this is something you have not to think about. So the replication, you would expect 95% in an ideal world, which normally never happens. So you're close to 82%. Also knowing that QM-Diab is a mixed ethnicity study with Arabs, Indians, and Filipinos, while Cora was a German study. And even under these conditions, we still replicate 82%. So I think that's really what I said before. Also with a high CV of the SomaLogic platform, it's very reproducible, very robust. When we did this study, there were not so many other PGWAS with SomaLogic out there. I mean, with SomaLogic, I think there was only one smaller one. So we had to go for replication also using other techniques and platforms. So there were a few PGWAS out when we did this study, but they were like immunoassay ones. So you see here, for instance, there's one, they had only 8 cis pQTLs. We replicated 2 out of 4. There was one very early SomaLogic studies where they only had 100 samples. So we almost had 3/4 replicated from that other SOMA study. There was a mass spec study, immunoassay study. I think there's Olink included here, so that's another platform, and then also MS study. So you see there's pretty good replication even to other platforms. And then the other amazing thing is if you go to the GWAS catalog, you would find GWAS papers on granulins, GWAS, and von Willebrand factors, and all of these GWAS in the GWAS catalog. They are done one by one using immunoassay platforms. And what we do with one single study here, we actually replicate all these 16 different GWAS on just one platform. All right, so this to sum up what the GWAS is on its own. So just to come back a bit, what we actually learn about this. So I mentioned already the term cis pQTL and trans pQTL. So a cis pQTL is an association with a protein which is actually encoded at the region where the variant is. Well, of course, I mean it's a kind of definition. What is a cis? Is it you take a megabase of distance? Do you do LD? But I think it should be pretty clear and not to be too dependent on the definition. And then you have another type of association. They are really interesting. And I mean, you have the same thing when you do transcriptomic GWAS as well, is a trans QTL, where there would actually be a variant that is far away or even on another chromosome from the protein encoding. And these kind of associations actually give you a network of actually protein interactions, if you want, not necessarily in physical interaction, but how one gene or the protein encoded in one protein at one locus actually interacts and influences the other one. And there could be things like, there could also be variants. And I think we have examples of that, like microRNAs being involved. There could be a variant that would regulate the expression of another protein. There could be protein turnover could be impacted by variants. The RNA turnover could be impacted by variants. And then, of course, you could have coding variants, stop codons, things changing amino acids or other microRNA binding sites. So I think there's a huge array of potential causal variants. And the challenge is, and also the interesting thing is now to understand what's really going on here. And if these things overlap with disease GWAS, I mean, then you're really onto something interesting. And I will come to that in a moment. But maybe before that, I should also go a little bit into the caveats. So if you have a bit heard about SomaLogic, I mean, the community is a bit split about them. Some people are a bit more critical than others. I think there's criticism, it's valid, but it's not valid in that it applies to all proteins, but to a few of them. So there's for instance, this kind of paper, and I think there are two things. The one is what we call the epitope effect. So the epitope effect, so what you see here is the betas of a GWAS with SomaLogic here with Aptam here, and the same GWAS using the Olink platform, which is a antibody-based affinity proteomics platform. And what you see is the betas of the GWAS pretty much align, which is very good. But then sometimes you find things like this, ICOM1, which actually is significant on both platforms, but with an opposite sign. And that initially makes no sense. But if you look into detail, now I have measurement on both platforms in our QM study. So here's the Olink data, and here is the SomaLogic data. And if I cross-plot them, if you wouldn't look at the colors, you would just see a blob here. But if you color them by genotype, then you suddenly find that there's actually 3 parallel lines. So what actually happens is you have like an here. So here's the association. So there's a negative association here with this genotype in SomaLogic and a positive association in Olink. And what actually happens is that there's probably one what do you call an epitope changing variant there, which actually interacts with the binding of the uptime. So we would have different binding efficacies for that, and then individually with the genotype, they all 3 correlate. And best correlates the AA, so that's probably the most strongly binding. So to wrap things up, epitope effect is actually what we would call genetically influenced binding. And I think it's important to think about that. SomaLogic actually doesn't measure protein levels. It's measured actually binding, like how much do you bind? If the binding becomes genetically influenced, then you get a signal, but the signal is not meaning there's different protein levels, there's different binding. And you have to be aware of that in the interpretation. So indicators for epitope effect are when the effects are very strong in cis, and especially if there's no overlapping QTL with the expression data. And then, of course, you could check whether there's a protein variant in LD with that variant, and then you would have a candidate for that. Remedies would be, I mean, you could confirm that with other assays as we did here. We could correct for the cis-SNP once we identified that. And then, of course, go for eQTLs for co-localization. And also, we now have a study with whole genome sequencing where we could actually check whether there is any variant around there. And what turns out, I mean, I don't have this on the slides, it's not published yet, but the numbers is a bit like 10% of the proteins actually have a coding variant. And of these 10%, still 5 of them overlap, 5% overlap with GWAS catalog hits on diseases. So basically, I would say there's maybe 5% of the ones that are really pure epitope effects. And I mean, they're still biologically meaningful. There's really a change in the protein. It's just not the protein. The second challenge is off-target binding, because that's a problem of all affinity arrays assays. Of course, you don't know what they bind to. You train them to bind to the protein, but for instance, here is a change log from SomaLogic, and if I just blow this up, they mentioned here for one protein that actually what they called alkaline phosphatase was actually lactotransferrin, because the protein they bought to train their somameres was not what was on the label. And then, of course, you can think about other things. There could be homology, things like that. So there are activities going on now in the field to actually validate each and every somamere using mass spectroscopy, pull down experiments, and things like that. And that's the kind of thing you would get if you do SomaLogic experiments and papers. Reviewers would come up with these two kind of criticism, and you have to, and that has to be moderated. I mean, here are 4 proteins out of 1,000 that are affected by this. So here again, you can look at this. So a strong trans pQTL without a cis pQTL, that could be an indicator. Because then the candidate at the trans locus could be the true positive. And as I mentioned already, so this is something that is now confirmed with pull-down experiment. We would actually pull down the protein using the uptermeres and then put them into a mass spectrometer to confirm. And there's a recent paper in Nature Medicine from SomaLogic where they actually in the supplement, they have like done this on 20% of the proteins already. All right, and then here maybe an example of something where you would think about the epitope effect, and actually it's not. So CPNE1 is this is a very strong one. You see here in these homozygotes for the TT, they basically have no CPNE1 levels in their blood. You would say, what is this? It's really very, very strong. And we can confirm this if we go into lymphoblastoid cell lines. So this is just public data, and we look at the transcription, it actually looks the same. And that's what I meant also before. If you want to confirm that something is not an epitope effect, if you find a transcription and eQTL corresponding to that, that of course indicates that there's a very good chance that the QTL is actually driven by expression. And for this one, we looked into it. We have RNA-seq data for these. And this is the aligned RNA-seq and around the CPNE1 gene. And what you see here is actually a variant in the transcript. And what you see is there's very rarely the C allele expressed. So this is allele-specific expression. So this is CPNE1 for a heterozygote of this variant. So one here. And what you see is actually all variants almost all come from only one from one strand. And this means that probably this one is a regulatory one, where somewhere one of the two alleles is suppressed. Maybe there's a promoter variant or something like that. And I mean, there's also a good example. It shows you what you can learn. I mean, with this kind of data, you can learn something about the function of the regulation of this gene. Okay, now I have a few examples. So of biomedical interest to show you a bit what you can do with them. I mean, there are a lot of paper. In the end, I will give you a list of them that you can use to, I mean, a lot of examples where what you can do with these pQTLs. So here, one of our early papers. So there are actually 2 pQTLs that associate with ankylosing spondylitis. And that's ERAP1. And what you see is basically the sum of the alleles. So the sum of the risk alleles between 0 and 4 of these two variants. And you see almost linear association with the allele count. So the number of risk alleles. And then here again, we have the lymphoblastoid cell line. So we can show, in this case, also, look, it's not an epitope effect. We have the same on the expression level. And these actually the risk of ankylosing spondylitis is also proportional to the number of risk. So with this one, we can now learn something about ERAP1. So ERAP1 is actually a drug target. And there have been discussions going on whether actually it's through genetic variation in that protein varying, or whether it's the expression level. What we see here, definitely it's the protein level. So if you would knock down the protein level, you could do something against ankylosing spondylitis. I mean, with everything that pharma then has to do to confirm the target. Another example here, SLAMF7. So SLAMF7 is the target of elotuzumab, and that's a monoclonal antibody for multiple myeloma. And what we find is that actually there's a genotype-dependent presence of this protein. So if you now imagine that you want to target SLAMF7, if people have different levels here, you would say that is probably, and confirmed also by expression, there's probably there's a risk of a genotype-dependent response to elotuzumab. So that would mean if you want to now treat cancer patients with this drug, it would be important to know their genotype. And I mean, it has still to be confirmed whether that plays a role, but there is a good chance that maybe the people with the AA allele are less likely to respond to the drug because they have already low levels of that. And then interestingly, you can also go for rare variants. So this is again the one you saw before, but there's a second variant, a rare variant, and that variant here actually totally abolishes the protein, and it also replicates in human diet. So that means that these people, they don't even have the protein in their bloodstream. So there's a good chance that these would certainly not respond to the drug. Other examples, I mean, this is a paper you may have seen already by Robert Plengy and co-workers about validating therapeutic drug. So it's really a very recommendable review, although it's already a bit old, but it's basically what pharma industry is doing nowadays with all these QTLs. So what they say is they use naturally occurring genetic variation to derive dose response curves. They would a bit look like this if you have different alleles. And we have examples here. So here, for instance, there's a combination of two different variants in IL-18. So I just have proteins here that are drug targets. So you would actually have some kind of a dose response curve here for that. There's another one here, and here's IL-6 receptor. So they are all examples. I think nowadays you would also come with Mendelian randomization of these. So these are actually instruments for Mendelian randomization. All right. Okay, so then let's maybe finally move towards the annotation of these associations. The other thing is, I mean, as I said, I already worked on metabolomics GWAS before. The thing is, when you do GWAS, you end up with a list of variants associated with lots of phenotypes, metabolized proteins, any multi-omics. But I think the trick is, if you really want to make sense of this, it's about how do you annotate them? How do you bring them together? How do you make sense of that and not just having a p-value list? So we have a tool which is called SNIPA, which actually does something that we call block annotation. So what you would see here is an LD block, different variants, different levels of correlation. And this would be the lead SNP or the query SNP of your GWAS. Then what SNIPA does, and I think there are other tools for that, of course, as well, it actually looks into, it actually translates information from GWAS catalog, from metabolomic studies, from GTEx, eQTL studies, or from ENCODE, and actually merges all this as one information together. Because in the end, if they are all in highly correlated variants, any of these is as good as a candidate for the causal SNP than the query SNP. So what SNIPA does, it does this block annotation, and then you actually bring together all the information you have on that locus to find out if there's something interesting going on. And doing this, we actually used SNIPA to bring things together, like the method overlapping methylation QTLs, regulatory ones, and you see the numbers here, like overlapping QTLs, eQTLs, transcription modifying variants, and then here are so the cis and the trans pQTLs. We have some glycosylation proteins. I think I mentioned protein modification, metabolites. That's interesting as well, because initially I was thinking there could be more overlap between metabolite and protein GWASs, but actually the overlap is extremely small, because even if you look at these 14 metabolites here, they are from the Metabolon platform, and they are not actually all metabolites. They are sometimes small peptides. So what this shows in a way, this non-overlap between metabolomics and proteomics QTLs, is also that there's, I think, a lot of complementarity between the platforms. So if someone would now come and ask me, should I do metabolomics or proteomics? I think my answer would always do both. And then in the end, what you really want to know is the association with the disease. So in this case, for all our hits, we have 83 associations for 42 different disease endpoints, which are here. And then you can put them into a network. This is something, I mean, systems biology is like and biologists hate is just like these hairy balls. So you just have like, you would have like your 500 hits here and your thousands of proteins, and then of course your 80s of disease endpoints from the GWAS. And then the thing is, how do you make sense out of this? And then you can zoom in. And what I like is now you can go and pull on things. You can just say, I'm pulling on a locus, I'm pulling on a disease, I'm pulling on a protein, whatever interests me. And we have a web server that provides that. So here's an example where I now pulled this and ordinated it. So this is an example where we would have like the ABO gene, so the blood group gene. What you find here is there are actually 3 variants. So the numbers here, I told you we have 400 over 400 loci. These are the numbers of the loci. On top are the proteins that associate, that are the blue lines. The gray lines are partial correlations between them. And to the bottom is what comes from the GWAS cutoff. So this is like association. You see there's tumor biomarkers, Lp(a), dipeptides, ferritin, coronary artery disease. And then I can go and annotate these. So let me just show this. So it's the same thing, just with more on it. So if I now go and annotate this, I mean, for this, I just used IPA. So I said, just say, tell me whatever you can find about these proteins and how can I connect them. Then, for instance, you find things like, let me just see here, for instance, here, Notch is Taiwan. Notch regulates Taiwan. So you can go to IPA, look up the actual paper. So this is not just like networks on like non-specific ones. So these are all annotated things. I found them using IPA, but they are all research papers. So Notch 1 regulates Taiwan. So you can Google it. There's a lot of stuff on that. Here would be here KDR associated with CDH4, KDR, FDL are heterodimers. Here MET, KDR form complex. So all these links are actually true links to them. And you see the GWAS brought all this together. So without the data gave you this. The literature here is something that people have found out before, but it's all supported in a way by the data. And if you put all that together, in the end, you come up here with plays a role in angiogenesis. So what this together says in a way that these genetic variants probably here with these proteins, there's a lot of angiogenesis around. And then you see, I mean, ABO is a risk locus for, or blood type is a risk factor for cancer. So I think it would explain a lot of the things of the pleiotropic associations of the ABO locus with different things, with cardiovascular disease, cancer, and so on. And based on this, now you have all these proteins and you can draw your conclusions. I mean, you see here also the A and the O alleles. All right, so this just as an example. Now, if you really want to work with this, so I have a nice team working with me in Germany, and we have a server that's called proteomics.gwas.eu. And I won't show you more details here, but if you go for that web server, you could actually click on all the individual links and just go through them. You would get the networks and be able to have all the SNIPA annotations on them. All right, so with this, I come to my conclusions. The title of my talk was actually the one of a review paper we recently wrote, Genetics Meets Proteomics, Perspectives for Large Population-Based Studies. And if you're interested, so here this red cube link, it's a Nature Reviews paper, but it's not really behind a paywall, because with this link you can get access to that paper. The central idea, and what I hope I showed you today is summarized in this central figure. I mean, you've already seen the idea that these proteins are circulating in the bloodstream. We learn a lot about the genetic variation in them. What I didn't discuss too much, I mean, I showed here, hang on, this is here. So the middle, no, the bottom one is supposedly be the uptime here, the homologic technique. But of course, there in the paper, we discuss other techniques as well. Here's the immunoassay, the Olink technique, and also the mass technique. So at the moment, there's a bit of a discussion, what is the best technique for scaling proteomics GWAS up in bigger population studies. And we discussed the different pros and the cons. Then in terms of analyzing the data, I think I've shown some of this in my talk, I hope was clear. So you would have like this kind of pQTLs, different genotypes associated with different levels of protein levels. And then here would be like a plot of the position where you would have the QTL and then the protein where it's encoded. The red diagonal would actually be the cis associations, and the off diagonal one would be the trans. And then I think things I didn't go into too much is, of course, nowadays that you have so much data, you have to look into co-localization as well. So I have to make sure if I overlap a disease hit to a protein hit, that they're actually the same genetic signal. So that's what is indicated here, that you have to look for co-localization. Is the regional association, but does it have the same shape or not? Analytical considerations, I think that's what I mentioned with the epitope effects, the binding specificity. There are similar things that might happen also with mass spec. And then in terms of applications, let me just see. What I didn't talk much about here, but I think that would be a talk on its own, is Mendelian randomization. So I can do a lot of MR with this now, drug target validation. I've shown you drug purposes, safety and repurposing. We have examples. So for all this in the review paper, we have examples. The network work, I think there are a lot of stuff you can do with network analysis there. One thing we did, and maybe that didn't come up too much, actually there was Anderson et al. Like 10 years back, they looked into all the proteins that are FDA approved protein analyzers, and they came up with 100. And we just redid this analysis here. They actually doubled, the number doubled in the last 10 years. So there are 200 FDA approved protein analytes now that you can measure. So that's a list here. And what's interesting is we can now check which ones of them actually have a pQTL. And what we find is that almost a third of them have a pQTL. So that means all these FDA approved markers are actually overlapping. They may have a lot of genetic variants, sometimes big ones. Okay, so finally, and if you want to dig deeper, so I'm maintaining a table now with all the GWAS with proteomics. That's on metabolomics.com. So sorry, I could change that to proteomics. So they are now, I don't know, I haven't counted. I think there are about 40 or so papers now. So the sizes are raising. So there's I think if you want to have the reference, people always talk about the Sun paper, where they had 3,000 samples with a bigger in-house platform from SomaLogic, almost 3,000 proteins. And then there's also a Science paper from the AGES study, where they had even more samples and more. And then recently, this is with the Olink platform. So there are now 30,000 individuals, but you see there's only 90 proteins. So that paper you will all find them in this list here. And where are things going? I mean, that's the latest press release from UK Biobank. So they are now aiming to do the full Olink platform, which would be 1,500 proteins by now. And they consider this a pilot with 53,000 biobank participants. So the aim is, of course, to go for the full biobank, which would be 500,000. And given what you've seen already, I think it's really pretty amazing what could be expected from them. All right, and with this, I think I have to acknowledge, of course, my founders. So I'm funded mostly by Qatar Foundation under the BMRP project. I could not mention everybody, but these are my present and previous team members. There's a lot of contribution for different labs here in Qatar and also around the world, especially in New York, in Munich. And with this, I thank you for your interest. Hope things have been clear. And if you have any questions, please let me know.
Thank you so much, Dr. Suhre, for this comprehensive review. It's been a pleasure. If anyone have a question, can speak up or leave a comment in the chat box. Meanwhile, let me start with a question. That's a more general question. With all these recent advances, which one do you think would make a major breakthrough in personalized medicine in the near future, such as the better facilitation of the trans pQTLs or extending pQTLs to rare alleles or anything else? What do you think?
Well, I think they are going in multiple directions. You mentioned rare alleles. I think why pharma is investing into UK Biobank is they want to find another PCSK9. So PCSK9, they have this kind of rare knockout families. They have very low LDL levels, and they found that this is a very good drug target, and they make blockbusters out of this. And I think that's really the thing. I think in terms of pharma, it would be the natural knockouts with extreme phenotypes. That's what they are after. Of course, that's not really personalized medicine. That's just drug target validation. I mean, I've shown you the variants that are in these FDA-approved markers. The point is, of course, if you want to do precision medicine, of course, you have to genotype people before you can do something with that. So I think in terms of personalized medicine, finding variants that play a role, you've seen the SLAM-F7, for instance. I mean, that would be personalized medicine. If you know, you have to look for this variant if you want to give this drug. So I think on that end, there's a lot that can be mined out of these pGWAS, especially if they become larger and larger and more powered. Otherwise, I mean, the definition of precision medicine is also a bit something I'm sometimes struggling with, because I'm also, you probably know the Englander Institute for Precision Medicine at Cornell. So for them, precision medicine is basically cancer. So targeting cancer based on the genome of the cancer. When we talk about precision medicine more here in Qatar, it's much more diverse to say, I mean, how would you do precision medicine for diabetes, for instance? Is it really only the genetics? Is it the metabolomics? There have been recently approaches to subtype diabetes into insulin resistant, insulin dependent on different phenotypes like HOMA-IR, HOMA-B, and so on. I think a Swedish group did that. So I think precision medicine is not necessarily only on genetics. It could also be on classification that you could base on metabolomic profiles, on proteomic profiles. How can you classify people into groups? But of course, you need big studies to show which classification really makes sense, right? I mean, just classify people for classifying them is not useful. But if the classification gives you a hint on how to treat people, for instance, what I mentioned, this diabetes classification, they have one group where they're just having a clinical trial in Sweden on SGLT2 inhibitors. So you might then decide on giving SGLT2 inhibitors on people who have a specific profile. I mean, you would probably not go do metabolomics broad on these people. You would look at 1, 2, 3 parameters in the end. So in the end, it comes down to biomarkers that you would then use for precision medicine. And it's a bit the same thing for genotype things as well. You would always have a few variants that you would really use for your decision making, unless, I mean, of course, if you talk about polygenic risk score, that would be another precision medicine. So that's a bit, I mean, I could rant on about whatever is there, but that's what I would have to say to that. For me, is a bit like all these omics technologies, it's a bit like imaging. So the more detail, the more pixels you have, the better you can see something. And once you see things, you know things. And once you know the things, you don't need the image anymore. So it's, in a way, it's something that brings you somewhere. In the clinic, I don't think you would use full proteomics or metabolomics in the clinic. You might, but at the moment, I don't see that coming very immediately.
Thank you. Any other questions from the audience?
I have a question. Great talk. Yeah. I'm wondering about the cis pQTLs. Are they replicating better than the trans pQTLs?
I think in principle they are, but partly also because they have more power. Because the cis ones, I mean, they are more direct, right? If you have a variant that directly changes the expression or even changes the proteome form, the effect is much higher. A trans one would always be through something, so you have always more noise on them. And I'm always a bit wary about the very strong trans. The very strong trans are a bit suspicious in my mind. They could be off-target effects. They could actually be cis where you have the wrong label.
Okay.
But there are all kinds of, I mean, that's an ongoing discussions. I'm always a bit frustrating about reviewers just brushing them off. That's why maybe I have a bit more slides on this, just saying, oh, you cannot believe the thing, don't do it, don't trust this thing. They just brush things off. I think you have to take this thing with moderation and know and be cautious about what it is and not just jump to conclusions. But I think they give you a lot of information you can work with. And even, I mean, even if you have cross-reactivity, for instance, if you're an aptamer that would target a protein and there would be a homolog or paralog in the genome, of course, there is a chance that it binds both of them. I mean, these are things you have to figure out. The interesting thing, there's so many things in these aptamers. Sometimes they're not targeting one-on-one a gene. They're targeting sometimes protein complexes. So they are aptamers targeting like the complexes of 2 or 3 proteins, and the aptamer might even bind between them. So it would not just target one. So it's even hard to say, is it a trans or is it a cis hit?
Okay, and do you know, can you know for each aptamer what exactly it's binding to and which part of the protein, or you cannot know?
Yeah, I mean that's a challenging thing. One thing is just to see whether it's really binding. What it is supposed to do is the pull down experiment. So you just take blood and you pull down with your aptamer a protein and you see what you pull down, and then you put it through a mass spec and you see what you find. Do you find one protein? Do you find several proteins? Do you find the one you want? So that's that. Yeah. It's I mean, if you want to know where it binds, you have to do crystal structure. And I think that's not realistic. I mean, I've shown you some structures at the beginning, but they were from the early days of aptamers to actually show that it works. And of course, you can do a crystal structure of them, but the effort it takes to do that is just immense. I mean, if you really have a very valuable aptamer that you would want to translate into a clinical biomarker for an assay, where you just use one aptamer on an assay, I mean, then it might be worth it. But otherwise, maybe what I should also mention here, I mean, in terms of using things for clinical application, SomaLogic actually now had the Nature of Medicine paper I mentioned. They now also do machine learning to actually train like hundreds of protein outcomes in parallel as predictors of whether you get a second heart attack, things like that. So that's actually their present business model. So they want to have this kind of what they call liquid biopsies. So you would just give a drop of blood and they would run all the somomers and they would have trained them on big studies. I think they have financed quite a number of them. And then they would machine learn them on whether you get A and B and C, and then give you scores on that. So that's maybe another way of how people could see precision medicine. What it's worth, I mean, you have to figure out. And then there's different applications they were talking about. It's not just like for predicting risk. It could also be very helpful in clinical trials to see whether outcomes change and things like that. And that's also, I mean, maybe anecdotally to know, I don't know how much you know about SomaLogic, but there has been some drawback in the last year. So we don't have a SomaLogic platform anymore. And the reason is that SomaLogic at some point changed their strategy. Initially, they were a bit like heading like Affymetrics. We have an assay, you pay for the assay, you measure, and you just do what you want. Then some people showed that there could be very valuable biomarker found, and then they got a big investment, and they said, no, we don't want other people to patent away what we measure. We don't want to be a service anymore. So they discontinued the kit and only did only under a license agreement measurements. So that's why we don't have them anymore. So a lot of the community actually moved towards Olink. They have changed in the past and now they move back. So they are back to the model, not the kit model, but the model of not taking away IP and then doing more fee for service. So I think that's a good choice. See how that works out. But there's certainly a lot of business behind all this and a lot of interest.
So now for a normal lab that wants to do a pQTL study, what technology is the most accessible?
Well, I mean, SomaLogic offers, again, fee for service. So that's what we did with Cora. I mean, we measured the QM-Diab and other studies in Qatar, but the Cora was fee for service we sent to SomaLogic. So you can send your to SomaLogic, pay them on a fee for service basis. You can do the same thing with Olink. And you can do the same thing with different core labs. Unfortunately, not the SomaLogic anymore. Before, I think the NIH had funded a few labs. I think there's one lab with the Gerstein in Boston who have done quite a number of measurements. But I think now that they don't do the kits anymore, all that is stopped. If you want to do a pQTL thing, I mean, the question is really what you want. Do you want to have the hugest number of proteins? Or the thing is, if you do smaller studies, it could kill you because the multiple testing would just kill you. If you do more clinical studies with 100 samples, 200 samples, then you don't want to measure 5,000 proteins because you wouldn't get any significant signal there. And for that, sometimes the Olink platform with just measuring targeted set, they have this kind of panels that just measure 90 cardiovascular proteins, 90 single proteins, things like that. That could be more advisable at that level. But it's an interesting field to follow, and I wouldn't say a last word on that. It's best is, of course, have both and to check them against it. I get some for them. Also here there are new technologies coming from.
And for the cis pQTLs, you showed some really nice examples where it's the same as a eQTL, right? The same effect. Did you also find some cis pQTLs where it's clearly like a different effect from the eQTL? And if so, where?
No, definitely. I mean, you would not expect an eQTL behind each cis QTL already, because you don't measure the same compartments. If you do a cis eQTL, normally that's expression data from white blood cells. The proteins are from the blood. So if the proteins you're measuring are coming from the white blood cells, then I would expect them to overlap. But if they come from elsewhere, they don't necessarily have to overlap. Might not even find a cis QTL because you don't. I mean, in GTEx you have them in other tissues as well, and then of course you could have things where where actually the protein level is not regulated by expression. It could be regulated by stability of the RNA. It could be, I mean, of course, the epitope effects. There could be the stability of the protein could be influenced, and so on and so forth. And there could also be feedback. I mean, there could be, like a protein could be more efficient because of a mutation, and then it could be back-regulation so that the protein levels are down-regulated again, or the expression levels are up-regulated. So I think you can imagine all kind of feedback loops there as well. So I think I would say if you find something that overlaps, it's always confirmatory. But if you don't find it, doesn't necessarily mean it's not there.
And have you found any variant that you could hypothesize that it's affecting translation levels? So you don't see the eQTL, but you see the pQTL. Or you haven't gone that deep. Maybe you don't have the power.
I mean, there's one variant, that's the IL-6 receptor. I mean, that's actually a very well-known variant. It just influences the cleavage of the IL-6. So that definitely is not, there's no eQTL behind it. The IL-6 receptor is expressed everywhere, but then whether it's cleaved or not is the QTL. And then the somologic targets the soluble form. So you actually what is solubilized, which in the end is driven by the variant in this kind of cleavage site.
Okay, interesting.
And in terms of, yeah, I mean, translation. I mean, they had at some point, they even had aptamers for ApoE variants, but I think they discontinued that. I don't think they were really very, very specific. I tried to figure out whether there was, I mean, there was some kind of signal, but it was not really conclusive. So they have sometimes multiple aptamers for the same protein, and they would bind at different places of the protein. So theoretically, you could also have readouts for protein modifications and things like that. I think that's a bit also where they're heading. But of course, the problem is, how do you train these aptamers in a concise manner?
Yeah, I actually have a related question, but I think you answered a lot of it. I guess Maria is kind of referring to the PSQTL paper by Alexis Battle, and it will be interesting to see protein-specific effect. But I want to ask, I guess, the other direction, because you have a lot of experience analyzing data from different population. So how do you deal with data from different population, and have you found, how do you see like QTL patterns to be specific or shared across populations?
I mean, we're just at the moment working on a proteomics GWAS in the Qatar Biobank and just trying to get a handle on that and to find out, is there something interesting there? We recently published a GWAS, a normal GWAS, on clinical traits on Qatar Biobank. And there you can already see, if you take polygenic risk scores from one population and from the other, you can already see that there's a difference. And I think also the recent paper from UK Biobank on clinical biochemistry, they already show differences in how well polygenic risk scores fare when you try to predict any of these traits. And of course, you see a bit the same thing in the populations as well. If you compare the Qataris and the Europeans, Although I must say, I'm a bit surprised. I think the Qataris are not so different from the Europeans. I mean, they are Arabs, but I think it's not like Africans in terms of distance. I think they are closer to the Europeans than to the Asians, for instance. But the tricky thing with the Qatari genome is it's not only that they are, I mean, they're really from a melting pot here. And then you have a lot of cousin-cousin marriages. So there's also a lot of inbreeding and things like that in the genome to be found. So we find rare variants more often than you would expect and things like that. I mean, rare variants in the homozygous state.
Thank you.
So, I mean, that's something we're working on. For the QM-DIAP study, it's 3 ethnicities, but there's, if you look at the size, it's a bit like 120 people from each ethnicity. So that's not large enough to really do lots of stuff with that. The interesting thing is you even find on the metabolome, you find things you would never, or you would expect them, but you would not expect them to see them in the data, like nutritional differences. Like we find metabolites specific to fish between the populations, which are clearly different nutritional habits of the people who live here in Qatar and things like that. It's really pretty amazing to have all this omics data together and to see what it does. And I mean, the genetics are only one part of it. What I didn't talk about here is the epigenetics. So we also have GWAS on proteomics. And in my view, it's a bit like the GWAS is a bit like it represents the lifestyle factors and the exposure. So in a way, if you go into this nature and nurture question, in the end, what you find is that you have like the genetics that defines one part of you, and the epigenetics, it doesn't define, but it's a readout for the other part, like the modular, the lifestyle factor part of you. And that's something we see in the metabolomics and also in the proteomics, so that you can actually do GWAS with these omics. That's a coming field, I think, and a much under study field, where you also have a lot of prejudices, where people just think too simply like, oh, methylation isn't interesting, it's confounded with everything. I think the exciting thing is actually the confounding, because the genetics is easy, because you know the SNP is always causal, because it has been there when you were born. But if you go for epigenetic studies, you never know when the methylation has been changed, why it has been changed, things like that. That makes it very complicated, but at the same time, it carries a lot of information. I think for data scientists, it's just a bane to go into that and to figure out what's going on.
Yeah, it's exciting for us.
Yeah, no, I mean, that's just the thing. I think where people tend to put things as negative, you say that's not negative, that's information, it's data, it's things you can make sense out of. So that's what I have to say.
Impressive. Any other questions from the audience?
Otherwise, don't hesitate to send me an email if you have other questions.
So if there are no other questions, please thank Dr. Suhre, who gave us a comprehensive review of the recent advances in genetics and proteomics technologies and how to interpret expression and protein QTLs and their limitations from a population space perspective. Thank you, Dr. Suhre.
Well, thank you, and hope to see you maybe one day in life.